Good day everyone, I am Dr. Sifache Basit and welcome to our lesson in Innate Immunity. Okay, so let us continue our discussion. So for this particular topic, we will be discussing the external defense system and the internal defense system. So this involves the internal defense system. We will be tackling on the pathogen recognition receptors, the acute phase reactants, inflammation. Let me fix my mic for a while. Apologize for that. Inflammation, phagocytosis, and the natural killer cells. Okay, so let's talk about the innate natural system. Innate immune system. Okay, so the innate immune system. Okay, the term innate means natural, or so therefore the other term for this is natural immune system, and the other one is non-specific immune system or non-specific immunity. So the defense against infection that immediately act when the host is attacked by a pathogen. Okay, so it is composed of two systems that work to promote phagocytosis. So number one is our external defense system. So this involves our anatomical barriers designed to keep microorganisms from entering the body. And then of course we have the internal defense system. So the internal defense systems include cellular responses that recognize specific molecular components of pathogen. Okay, so for this particular lesson, let's talk about the external defense system. So the external defense system is composed of physical, chemical, and biological barriers that work together to prevent infection from entering the body. Okay, so as you can see in the illustration here, so let's talk about the skin. So skin is considered to be as a physical barrier. So our skin is made up of keratinocytes, which produces keratin, and keratin in our skin will keep it dry. And of course, there's no water activity. It's gonna be difficult for microorganisms to grow or multiply in our skin. Not to mention that our sweat glands are actually producing lactic acid, kaya pala ma-acid minsan, and this will also have antimicrobial properties. And then the sebaceous glands produces sebum. Okay, sebum or the sebum or fatty acids, and this will also have antimicrobial properties. Okay, now biological activity or chemical activity would include lysozyme. So whenever you are crying, that's the best way to clean your eyes. Pag nagpapaiyak kayo ng mga girlfriend or boyfriend nyo, sabihin nyo, mahal na mahal lang kita, pinapaiyak kita, so that, you know, your tears would have lysozyme, and lysozyme would also have antimicrobial properties. So you can say that. Walay na naman yung joke ko, no? Anyway, your stomach. So whenever you have ingested something, chances are these microorganisms will not be able to tolerate the acidity of your gastric juice. And whenever you are coughing and sneezing, you are expelling microorganisms away. That's why social distancing is necessary. Okay, so the skin, as what I've told you, is made up of physical barrier. Okay, so when you say physical barrier, the secretions will discourage the growth of microorganisms. So particularly, your epidermis is made up of tightly packed epithelial cells coated in keratin, so that will keep it dry. And then the dermis is made up of connected tissues with blood vessels, hair follicles, sebaceous glands which produces sebum or fatty acids, sweat glands, and even with WBCs. Okay. The skin, lactic acid, and fatty acid will maintain a pH of approximately 5.6. So that's very acidic. And there's a particular protein known as psoriasin, which is produced by skin cells. And psoriasin would also have antibacterial effect. Okay? Now, let's talk about your respiratory tract. In the respiratory tract, the mucous secretions will block bacteria from adhering to epithelial cells, which means that whenever you cough, whenever you sneeze, you allow or you are actually expelling out pathogens from your respiratory tract. Not to mention that your trachea is actually made up of what you call this? It's actually made up of pseudostratified ciliated columnar epithelium and the cilia propel away the. And hi, here's my son Josiah. Hi Josiah, say hi to everybody. And you know, the favorite song of Josiah is Knees and Toes, Head and Shoulder, Knees and Toes, Knees and Toes, Eyes, Ears, Mouth, Nose, Larynx, Trachea, Head and Shoulder, Knees and Toes, Knees and Toes. Ayan. So remember, the trachea has pseudo-stratified ciliated columnar epithelium, and that will propel away, okay, the mucus. Okay? And of course, if you propel away the mucus, the cough reflex will somehow be stimulated. Say hi, baby. Okay, so that was fun. So let's now move on with our lesson. So urine, still part of the external defense system. The urine helps remove potential pathogens from the genitourinary tract. So whenever we are flushing away the urine, we keep somehow the urethra free from potential invading microorganisms. And speaking of the digestive tract, which I mentioned to you a while ago, do you know that the stomach's HCl or hydrochloric acid will keep the pH as low as 1? Can you imagine? A pH of 1 is even enough to dissolve, its corrosiveness is even enough to dissolve lead. So it will prohibit microorganisms from growing. So you might be asking me, how come, if that's how potent our stomach is, how come that there are still microorganisms? I haven't plugged the mic. Sorry. Okay. So you might be wondering, how come if our skin or if our stomach has an acidity of as much as 1, how come there are still pathogens that can cause diarrhea? Well, the reason, again, take example, Entamoeba histolytica. It's cyst. The cyst, which is the infective stage of Entamoeba histolytica, will allow them to resist the stomach's HCl. There are some bacteria that can even thrive on that particular HCl, Helicobacter pylori. And then Salmonella, E. coli, Shigella can still cause diarrhea by of course, by drinking contaminated water and eating contaminated food. Food and water serve as the vehicle for them so that they will actually escape the acidity of the stomach. Okay, so that's the reason why normal flora or microbiota will help us to keep pathogens from establishing ecological niche in our digestive tract. So there you have Lactobacillus acidophilus. That's in the vagina pala. Lactobacillus casei. Okay? Yung Lactobacillus shirota strain, the one that you can find in Yakult. So probiotics are important to keep our digestive tract healthy. Okay, so let's talk about the internal defense system. So the internal defense system includes the pathogen recognition receptors, the acute phase reactants, inflammation, phagocytosis, and the natural killer cells. Okay, so let's talk about the PRRs. Okay, the pathogen recognition receptors will allow us to recognize molecules unique to infectious organisms. So where can we find the PRRs? We can find it on macrophages and dendritic cells. Common denominators, macrophages and dendritic cells are considered to be as what? Antigen-presenting cells. Then neutrophils, eosinophils, monocytes, mast cells, T cells, and even epithelial cells. So you can see that majority of these cells, okay, are found, or majority of these cells would have the PRRs. So the PRRs are encoded by host DNA to sense extracellular infection. Wow, which means that our cell somehow would have this would have the mechanism of identifying whether there are invading microorganisms. Okay, so they can recognize the so-called pump. What is a pump? The pathogen-associated molecular patterns. Okay, when bound to a pathogen, there's a pump, then it will activate phagocytosis. Okay, so what are examples of PRRs? One of the most common examples of PRRs are the TLRs. Okay, so TLRs are known as the toll-like receptors. Toll-like receptors. There are about 10 types of TLRs found in humans. Some are found on cell surfaces, some are found in cytoplasm. So as you can see, the toll-like receptors can actually recognize pump. And when these pump are recognized, it will activate phagocytosis. Okay? So it can recognize motile bacteria, for example, gram-negative bacteria, fungi, mycobacteria, viral double-stranded RNA, viral single-stranded RNA, viral bacterial or viral DNA. So there are about 10 types of TLRs. Okay? TLRs are glycoproteins that bind to a particular substance. Okay? It can activate cytokine and chemokine production and other processes to enhance phagocytosis. So once TLRs are activated and phagocytosis have taken place, it will destroy most pathogens that humans are exposed to even before the disease sets in. So that's how important phagocytosis is. So aside from TLRs, there are other PRRs. So one is CLR. So this is the C-type lectin receptor. So this one is bind to mannan and beta-glucans found in fungal cell wall to activate cytokine and chemokine production. So mannans and glucans are found in fungus. So somehow CLR will most likely prevent fungal infection. And then we also have the RLR, or the retinoic inducible gene 1 I-like receptor, and then NOD, or the nucleotide binding oligomerization domain receptor. But it is not as extensive as the toll-like receptor. Okay, so do you know that whenever we get injured, infected, or whenever there has been an injury or tissue trauma, we produce a substance known as the acute phase reactants, a soluble factor found in our serum. So what is the purpose of the acute phase reactants? So somehow the acute phase reactants will facilitate contact between microbes and phagocytic cells. It will mop up and recycle important proteins after phagocytosis. So that's acute phase reactants. So the most common example of acute phase reactant is CRP. So it is commonly being requested by physicians or clinicians in order to assess inflammation, non-specific inflammation. So the response time is about 46 hours. Normally we have CRP, but as you can see here, CRP increases up to 1,000 times during inflammation, during injury. So the function is opsonization, complemented activation. Okay, but CRP, that's the reason why the CRP is the most popular, because as you can see, within 4 hours after you have been inflamed, CRP level in our body increases up to 1,000 times. Unlike others, you have to wait for 24 hours, or for ceruloplasmin and complement C3, you even have to wait for about 3 days, 2 to 3 days before you can detect it, and its level will simply rarely increase up to 2 times. Unlike CRP, exaggerated increase up to 1,000 times within 4 to 6 hours after the onset of inflammation, injury, or tissue trauma. So that's why it's actually being utilized. There is also what you call the HPCRP, high precision CRP, and it is also being assessed to evaluate acute myocardial infarction or heart attack. There's also another protein known as serum amyloid A. Its function is to activate monocytes and macrophages, but you have to wait for 24 hours, the response time. Then alpha-1 antitrypsin is a protease inhibitor. And then fibrinogen is important for clot formation. Then haptoglobin can bind with hemoglobin. Ceruloplasmin can bind with copper and oxidized iron. But ceruloplasmin is important because a deficiency of ceruloplasmin may result to Wilson's disease. And then complement C3 is important for opsonization and lysis. C3 is also important. It is the most abundant complement component. Okay. Speaking of complement, we will have a separate discussion with complement later. Okay? So, let's talk about the process of inflammation. So, inflammation is our body's reaction whenever we are injured or whenever there is an infectious agent invading our body. So, we react. And we react by redness, erythema, swelling, heat, and pain. So these are the cardinal signs of inflammation. In Latin, redness is rubor, swelling is sumor, heat is calor, and pain is dolor. So these are the cardinal signs of inflammation. So what will happen during inflammation? By the way, I will also be including an animation in this module. So be sure that you'll be watching that animation. So what will happen here is that resident macrophages and mast cells at the site of infection will release chemokine that causes vasodilation and induce lectin. So that's the first step. Okay, and then lectin will loosely bind circulating leukocytes, as you can see here, and cause them to roll along the vascular cells. And you know what's so important about this leukocyte is that, by the way, the pink cells that you see here, these are the endothelial cells. Okay, the endothelial cells are actually the epithelial cells that line our blood vessels. And because of the lectin, they will bind. And then, what can you see here? Leukocytes, the integrin particularly, will enable the leukocytes to crawl in between the endothelial cells. So since these are just simple squamous epithelium, leukocytes would have the ability to squeeze themselves out. So we call it diapidesis. And they can easily go at the site of the okay. So that's the reason why whenever you're inflamed, you'll see pus, meaning to say many WBCs. Pus, or in Tagalog we call it nana. It means that there are many WBCs that have actually gone through that particular site of injury or inflammation. So most of the time, the cells involved are, for phagocytosis, are neutrophil, monocytes, macrophages, and dendritic cells. But before I discuss phagocytosis, let me go back to inflammation. What's the purpose of inflammation? The purpose of inflammation is, number one, you do not want to have systematic infection. So you want to localize the infection. You do not want to spread further the infection. So number one. And then number two, for tissue repair, in order for you to heal. But before you can achieve tissue repair, you have to undergo the process of redness. Why redness? Because of because during inflammation, there is vasodilation. So your blood vessels dilate, more fluids will come in, so that makes somehow your blood vessels edematous, so they're swollen, redness, and because of all the activities happening around here, so there is an increased heat. And during inflammation, you will produce pain receptor, a mediator of inflammation such as prostaglandin. So that's the reason why you feel pain. However, tandaan natin, you need to feel, okay? So that's the purpose of inflammation. Kaya para maka-move on, somehow, kailangan mong pagdaanan yung proseso na to. At one of the processes that you have to undergo to in order for you to achieve healing sa mga kapatid, kung may nararamdaman ka. Okay, so moving on, let's talk about phagocytosis. So, phagocytosis involves neutrophils. So remember that the most important cell in phagocytosis is neutrophil, and then followed by monocyte, macrophages, and dendritic cells. Okay. So, in phagocytosis, first of all, the first step is adherence. Okay, as you can see here, adherence is the physical contact between the phagocytic cells and the microorganisms aided by opsonin. So remember, an opsonin is an antibody that mediates in phagocytosis. So you're not able to see opsonin here, but there are actual opsonin here. And then number 2 is engulfment. So as you can see, it is the outflowing of cytoplasm to surround the microorganism. Okay? And then number 3 is the formation of phagosome. So phagosome is like the organism once inside the cytoplasm of the neutrophil, for example, there is a vacuole. Okay? The vacuole here is called phagosome. Okay? Microorganism is completely surrounded by part of the cell membrane. And then, the organelle that you're able to see here, we call it the lysozyme. Okay? So the lysozyme is like the suicide bag for the bacteria because the lysosomal granules will actually come into contact with the phagosome and will fuse its content in the phagosome. Therefore, there is the so-called formation of the phagolysosome. So what happens here is that the content of the lysosome are emptied. So the lysosome emptied. So the component is called lysozyme. Okay? So what will happen? Number 6, you will see that the lysozyme, it's a hydrolytic enzyme, will digest the microorganisms, and eventually the microorganisms will be expelled out, okay, of the phagocytic cell, and you call that exocytosis. However, there are some microorganisms that are actually resistant to phagocytosis. Number 1, if they have capsule. Because capsule is made up of slimy polysaccharide layer, and that somehow will prevent them from being engulfed. So that's number 1, capsule. And then there are even some bacteria that can multiply inside the macrophage. An example of that bacteria is Mycobacterium tuberculosis, the etiologic agent of TB. Okay, so once the macrophage have ingested Mycobacterium tuberculosis will even multiply inside the macrophages. Ganun sila ka-active. Okay, so however, without capsule, if it's not TB, WBCs can engulf and destroy invading microorganisms. Okay, so let's talk about the natural killer cells. Natural killer cells are also considered as the first line of defense against cells that are virally infected and those that are infected with intracellular pathogens or tumor cells. These are collectively called the target cells. So, natural killer cells are also known as the large granular lymphocytes. Okay? Large granular lymphocytes that can recognize any damaged cell and can eliminate it without prior exposure to it, giving immune system time to activate an adaptive response of specific T cells. And so the natural killer cells are stimulated by exposure to cytokines. Okay, so we will be discussing cytokines later on. And when activated, it will produce additional cytokines or colony stimulating factors. It can influence innate and adaptive immunity. So, as you can see, natural killer cells, some of the target cells can inhibit them. So if inhibition is present, then naturally there's no killing. So if the cell here is virally infected, okay, if the cell here is virally infected and there's no inhibition, the natural killer cells can induce apoptosis of a virally infected cell. Or if there is an antibody-coated target cell, natural killer cells may also induce apoptosis. Apoptosis is the programmed cell death. So we do not want to have any inhibition of the function of the natural killer cell, so that we'll be able to achieve the apoptosis of target cells. Again, target cells are cells that are virally infected, cells infected with other intracellular pathogens, and tumor cells. Okay, so there you have it. I'll just be running down the summary so that you can screenshot them. Okay, so there you have it. I hope that you're able to study very well in this particular setup. Feel free to communicate with me if you have any clarifications or questions. This is Dr. Supachay Basit saying, God bless you and good day, everyone.